This lecture is on transmembrane proteins known as ion channels and transporters. As you know, the cell membrane is made out of phospholipid molecules, which consist of a polar head and a fatty tail. And these phospholipid molecules arrange themselves in a bilayer that allows most things to stay on one side of the membrane or the other. In particular, hydrophobic molecules, such as a lot of gases like oxygen and carbon dioxide, actually go through the membrane, but other molecules often don't, and in particular, ions or any charged molecules cannot pass through the phospholipid bilayer, and therefore the bilayer is a good insulator of electricity. So how do ions actually, or any charged molecule, actually gets through the membrane? There are two ways for ions to go through the membrane, and both of these are transmembrane molecules. One category of these molecules that allows ions to go through is transporters. Transporters actively move ions against their concentration gradients, and they can produce concentration gradients between the cytoplasm and the extracellular medium. The other category is ion channels. Ion channels are also transmembrane molecules, but they don't actively do anything. They just passively allow ions to diffuse, usually down their concentration gradient. And we will come back and discuss both of these, but I want you to remember that the main difference between transporters and ion channels is that ion channels have a pore in the middle, and that pore allows ions to go from one direction to the other. The ion channel really does not care which direction the ion goes through, whereas transporters have no pore. They actually bind the ion, change their transformation, and release it to the other side. Ion channels are made out of subunits. In this example, this ion channel is made out of 5 subunits, and that is typical for the ion channels that we're going to discuss. I'm going to give you examples that are somewhat different than this too. These subunits arrange themselves around a central pore or hole, and that pore spans the space between the cytosol and the extracellular medium. The fact that there are subunits simply means that an ion channel is a complex protein that is composed of several different proteins that have arranged themselves into one big complex. Each subunit is coded by a gene within the genome. So that's how a subunit is defined. A specific ion channel type doesn't always have the same composition of subunits, and different types of subunit arrangements can make the properties of ion channels different, and we'll discuss that when we need to. In addition to having a pore in the middle, ion channels have a property known as gating, which is opening and closing, and that's really a conformational change in this complex molecule that allows this pore to remain open or to close. And the second property that makes ion channels different from a simple hole in the membrane is known as selectivity. And that means that ion channels are only permeable to a single molecule type, a single ion type, ion species, or at most a few ion species. So that is typically the primary way we name an ion channel. So we call it a sodium channel or potassium channel, depending on which ion it allows through its pore. A sodium channel does not allow any ion other than a sodium to go through, and a potassium channel does not allow any ion other than potassium to go through. So again, the two properties that make an ion channel different from a simple hole in the membrane are gating and selectivity. Gating, as I said, is a conformational change in the ion channel molecule that allows this pore to be open, that's gated open, or closed, that's gated closed. Typically, the ion channel goes between these two states at random. So it goes to this state and then randomly jumps to that and randomly jumps back to the other one. But the amount of time that the channel spends in an open state or a closed state depends on other factors that we'll come back and discuss. In particular, one of these factors could be voltage. So channels whose opening and closing depend on the voltage across the membrane. So that is how much the inside of the cell is sitting at a negative or positive voltage with respect to the outside. That's the transmembrane voltage, and the channel can sense this transmembrane voltage because it has access to both the inside and the outside. When the voltage inside the cell increases, and that we call depolarize, then this channel opens and allows ions to go through. When the voltage decreases, it closes and it doesn't allow ions to go through. A channel that does its gating in a way that it is sensitive to voltage is known as a voltage gated channel. Most voltage gated channels open when the voltage inside the cell becomes more positive and close when it becomes more negative. There are a few exceptions, and again, as needed, I'll come back and discuss that. Ion selectivity, on the other hand, refers to the fact not that the channel is open or closed, but that this channel, this pore in the middle, only allows certain ions to go through. The channel does this by having a selectivity filter, and a selectivity filter is really a collection of amino acids in the protein structure of the channel. The protein structure part that is actually lining the pore, that allows particular ions to go through and does not allow others to go through. So again, in this example shown, this is a potassium channel, and it allows potassium to go through and does not allow sodium, chloride, calcium, or other types of ions to go through. There are, as I said, different forms of gating for channels. The one that I actually told you about right now was voltage gated channels. So some channels are voltage gated or voltage activated. There are other channels that don't care about voltage across the membrane at all, but they open only when the channel binds to a specific molecule. That molecule that binds the channel and opens it is known as a ligand. A ligand can bind the channel from the extracellular side, such as happens in neurotransmitter receptors, or it can bind the channel from the intracellular side in some examples that we'll see, for example, in calcium activated channels or in cyclic nucleotide activated channels that we will discuss extensively in the context of the visual system. There are also channels that are activated by physical changes in the membrane, such as stretch of the membrane. And these types of channels, for example, are important in somatosensory touch reception. And they do a conformational change by detecting physical changes in the membrane. So one way of categorizing channels is by how they gate. A different way of categorizing channels is by what ion they allow through. So this slide shows you different examples of voltage-gated ion channels. So all of these channels are voltage-gated. They all open by voltage inside the cell going up. But this type only allows sodium to actually move through the channel. This is a calcium channel, only allows calcium to go through. This is a potassium channel, only allows potassium to go through, and chloride channel only allows chloride to go through. As you see, the direction that is schematically shown is different for the different ions, but this has nothing to do with the properties of the channel. We'll come back and discuss what decides which direction these ions go through. It is not the channel that decides that. It is a different factor. Ligand-gated ion channels can also be selective for particular ions, but many ligand-gated channels are not that picky. So for example, a lot of neurotransmitter receptors, like the glutamate receptor, are permeable to both sodium and potassium, and sometimes to both sodium, potassium, and calcium, but never to chloride. So we call these non-specific cation-gated non-specific cation channels. Okay, they're glutamate gated, but they're non-specific cation channels. Here's an example of a ligand-gated channel that is a calcium-activated potassium channel. So the permeability is to potassium, that's the ion it allows through, but the ligand that activates it is calcium, and calcium binds to this channel, not receptor, channel, and when it binds, the channel pore opens and it allows potassium to go out of the cell. The cyclic nucleotide channel can be activated by either cyclic AMP or cyclic GMP. These are different categories of channels. And when it opens, it's also a non-specific cation channel in that it allows both sodium and potassium to go through. So they're distinct forms of channel gating. Once again, I told you about voltage-gated channels. I told you about ligand-gated channels. I told you about stretch-activated channels. But I also want to come back and mention a category of channels that are none of the above, and they're non-gated, so they're pretty much always open. Okay, they don't care about what else is happening. Typically, we refer to these channels as leak or passive channels. Okay, so these types of channels are quite important in producing the resting membrane potential, which we will discuss in another lecture. Channels, in addition to opening and closing their pore, which is the gating of the channel, could be rendered inactive. They get rendered inactive when the pore is open through a separate process. Voltage-gated channels, when the pore opens, often have a property that a part of the channel comes back and clogs the pore. This process is known as inactivation, and as we'll see, is an essential thing when we discuss action potentials. Ligand-gated channels don't inactivate, but when they open with a ligand, after a while, even though the ligand is still bound, the channel pore closes, and that is known as desensitization, and that happens with many, if not most, neurotransmitter receptors, and again, we'll see that when we talk about neurotransmitter receptors. So here's actually an example of that. This is a glutamate receptor. At rest, it's closed. Glutamate binds to the channel opens, and you see a current going through the channel, but the current doesn't remain up. It actually goes back down, and that's because the channel desensitizes. Even though it is binding glutamate, it closes. Now, sodium and potassium channels that are responsible for producing action potentials have a set of properties that are quite important. Sodium channels in particular open when the voltage inside the membrane increases. That allows sodium ions to flow through the channel, and they will flow from the outside to the inside. But these channels are inactivating. That means that a part of the channel actually comes back and closes this pore. So even when the voltage is up, the channel closes and ions stop flowing. So sodium channels, these fast sodium channels, only allow ions to go through, sodium ions to go through transiently when the voltage is up, and after that they just don't. In contrast, the potassium channels that are responsible for action potentials, these channels also open with voltage going up, but they remain open so long as the voltage is up, and they allow potassium to go out of the cell from the inside. There's another thing that's schematically shown in this picture, and that is that when the voltage moves up, sodium channels open first before potassium channels open. How we know about the properties of channels is through a method that was developed by these two scientists Nair and Sackman. This method is known as the patch clamp method, and it is a way of putting an electrode onto a cell and recording its properties. And it is very useful for recording single channel activity, and it's different from the methods that were being used before that and are still being used, known as sharp microelectrode recording. So in this case, a glass micropipette that is filled with a fluid is brought down to the cell membrane. A mild suction is applied that sucks in a piece of the membrane, and that, with some luck actually, there's a channel or a few channels that are inside this pipette tip. The pipette diameter is about a micron, and by sucking, a tight contact is produced between the pipette and the membrane. This is known as a gigaohm seal, because the resistance is so high that it's about 10 to the 9 ohm, or a gigaohm. Now, after this suction is applied and the seal is obtained, there's several things that could be done. One is you can actually rip apart the membrane inside the pipette and therefore make the inside of the pipette continuous with the inside of the cytoplasm, and this is known as a whole cell recording. Or instead of that, you can pull that piece of the membrane apart, and if you have a channel or a few channels stuck there, the inside part of those channels, the part that was in contact with the cytoplasm, now is in contact with the extracellular medium, and for that reason it's called an inside-out recording. And for doing an inside-out recording, you pull this out and expose it to air, and then you start recording. If you retract but don't expose to air after a whole cell rip, the membrane just closes and the outside of the membrane becomes the same with the outside extracellular medium, and therefore the outside of the channel is exposed to the extracellular medium, and this is known as an outside-out recording. Patch clamp recordings that are done through a single channel look something like this. So what you see is the current that is passing. So current is the movement of charge passing through the channel. So here the channel is closed, here it's open, closed, open, closed, and you can see that it opens and closes in different properties. This is a glutamate activated non-specific cation channel. This one is an acetylcholine activated channel, which has a closed state but two different open states. So the closed state is up there, open one is here, and open two is there, and different levels of current pass in the different conformational open states of this channel. At the bottom, you have another transporter, neurotransmitter receptor. This is a glycine-activated chloride channel. Here, the closed state is down here, and the open state is up there. And you can see the movement of the channel, the gating of the channel is kind of random between the closed and open state. So it's not like it's always open or it's always closed. It keeps moving between these states. Gating, therefore, because this movement is random, is quantified by the amount of time that the channel remains in the open state. So if you record the activity of the channel for a long period of time, there's a fraction of that time that the channel remains open, and that fraction in proportion to the total time is the gating probability of the channel. So the fraction of the time that the channel is open is the same as the probability of the channel that is open, or alternatively, you can take a lot of these channels. Say you take 100,000 channels of the same exact type, you freeze them at one point in time, and you count how many of them are open at that point in time. That fraction of a large numbers of channels that are open at any time is also the same as the probability of the channel being open, because channels are working independently of each other. So gating can be quantified by any of these factors, and therefore gating as a number is always something between 0 and 1 because it's the probability of the channel being open. If it's always closed, it's 0. If it's always open, it's 1. Otherwise, it's somewhere in between. Now, if you compare the current that flows through sodium channels with the current that flows through the potassium channels, just as you increase voltage in a step across the membrane, you will see what we saw schematically through the channel by looking at the current. So each of these traces is showing one sweep where we move the voltage from a negative voltage to a very high voltage and back. It doesn't have to go positive, it just has to go more positive than where we were holding it. When you move the voltage up, the channel opens here. It allows current through. You can see the closed state and the open state, and then it closes itself. You do it again, it opens, closes, opens, closes. Do it again, opens, remains open for a while, and then closes, etc., etc. In this sweep, it didn't even open. Now, if you look at the potassium channel, you move the voltage up, the channel opens but doesn't close until you move the voltage back down, except because it is opening and closing randomly or stochastically, sometimes it closes but then it comes open. The probability of it being open is very high when the voltage is up. Now, if you look at all these traces here, what you see is that this channel remains open pretty much until you move the voltage back down, not this one. The current through this channel is going in the direction that is positive because closed is down, and as you open it, it goes positive. This one is going negative. Closed is up and it's going down. We'll come back and discuss that in another lecture too. And finally, not finally, so this channel opens much, much faster when you move the voltage up versus this channel, which can have some delay to its opening. All right, so the other category of transmembrane proteins that allow ions to go through are membrane transporters. Membrane transporters are membrane-spanning proteins, therefore, that are involved in the exchange of ions. They don't have a pore, and they involve no diffusion, and they fall into two categories. One category we call passive transporters. The other category we call active transporters, and that's because they have to spend energy to move an ion from one side of the membrane to another. So energy spending transporters are also known as pumps. If a transporter moves ions in a way that it produces a net movement of charge, so it moves, let's say, more positive ions in one direction than the other, or it only moves a negative ion in one direction, then you call it electrogenic. And I'll give you an example which is coming up, and that's the most important example for us. So here's an example of a sodium calcium exchanger where it moves sodium in the cell and pushes calcium out of the cell. Here's a sodium potassium chloride co-transporter. It moves all three ions into the cell. Here's a this is a potassium chloride co-transporter which moves potassium and chloride out of the cell. This is a proton pump or a sodium proton. Proton is the same as a hydrogen ion, so sodium proton exchanger. This pumps out protons from inside the cell in exchange for moving sodium into the cell. And here is neurotransmitter transporter. So this neurotransmitter transporter shown here can be transporting GABA or dopamine, okay, not together with dopamine, GABA or dopamine, and together with these, it also transports sodium. The transporter that is most important to us is the sodium-potassium pump. The sodium-potassium pump, which I'll discuss again in a slide briefly, moves potassium into the cell and moves sodium out of the cell, and it's an active transporter or a pump. And that means that it actually uses energy in the form of ATP that it hydrolyzes into ADP. And by doing so, it allows this exchange. The sodium-potassium pump actively moves these ions against their concentration gradient. By doing so, it produces an excess of potassium concentration inside the cytosol compared to the outside, and it reduces the amount of sodium concentration in the cytosol with respect to the extracellular medium. Another pump that is quite important to us in the context of signaling and synaptic transmission is the calcium pump. The calcium pump moves calcium out of the cell by, again, hydrolyzing ATP and moving a proton ion into the cell. So the way the sodium potassium pump works is that one side of the pump that binds sodium is exposed to the cytosol. When it binds 3 sodium ions, it hydrolyzes ATP, which phosphorylates this pump. And by phosphorylation, it changes the conformation of the pump so that these sodium ions become exposed to the extracellular medium. The sodium ions are released and the pump binds 2 potassium ions, and at that point it dephosphorylates and goes back to the conformation it had initially, and there it releases 2 potassium ions inside the cell. So by hydrolyzing a single molecule of ATP, this pump allows a movement of 3 sodium ions out and 2 potassium ions into the membrane. Because sodium and potassium ions both have the electrical valence of +1, by moving 3 sodium ions out and 2 potassium ions in, you're effectively reducing the net positive charge inside the cell, and therefore this transporter or pump is electrogenic. It produces electricity. It makes the inside of the cell more negative when it is active.